And what I'm going to try to do in the next 25 minutes is give you a journey in inflammatory bowel disease. I'm going to talk to you a little bit about genetics, so starting with the human patient with the disease, and then tell you a little bit about what we've learned in the microbiome around this disease, and then try to end my presentation by finding connections between human genetics and the microbiome. So if you look back at inflammatory bowel disease, there was a review in the New England Journal of Medicine about 2002 that basically said there was a genetic contribution to the disease. No genes were identified. Based on family studies, there was evidence that there was a defect in the mucosal barrier. And there were clearly environmental events which were very loosely defined as exposure to smoking that protected you from ulcerative colitis or increased the risk of Crohn's disease if you smoked. And if you had an early appendectomy, you seemed to be protected from ulcerative colitis. If you fast forward 10 years, we've now identified many genes or genetic associations with this disease, about 160 genomic regions that are associated with risk of disease. And despite the expanding number of genetic associations, one of the emerging facts is that the number of pathways that are altered are limited in scope. We've also been, at the same time during these last 10 years, we've sort of complicated the immune system for us. We've sort of appreciated that there are many subtypes of T cells, but more importantly, paid attention to the innate immune system as being a gatekeeper for immune defense in whatever organ system or mucosal compartment that one might think about. And at the same time, our understanding of the microbiome in disease has changed. So up until the year 2000, the search was for that pathogen that caused the disease. But I think we are beginning to appreciate that there may be good and bad microbial products. There might be the community structure that might be important, and the stability of the flora might be more relevant in altering or recurrences of disease. It's been a collaboration of the Broad Institute as well as with Rob Knight, where over the last 2 years we've put together all the available methods to try to analyze the microbial community. Without going into detail, I'll talk a little bit about the pie crust method that was developed by Curtis Huttenhauer and Rob Knight that takes 16S sequences and tells us something about function. And then also approaches to profile the community. And then I'll also briefly allude to the Maslin algorithm that tells us something about biomarker discovery based on microbes. So the take-home message with inflammatory bowel disease, whether you look at children with early onset disease or adults with established disease, most of these studies are snapshots of disease. There's clear evidence that the diversity is reduced. There are specific clades that are either over or underexpressed depending on the disease state. And it's likely that the microbiome will determine the definition of the subtypes of IBD. So unlike infectious disease, the emerging concept in many of these complex diseases, from IBD to diseases such as psoriasis, is that there may be many microbes that initiate this response. So if you apply this tool, multivariate analysis, to begin to understand what are the microbes, so what is the abundance that's seen in patients on various treatments. So mesalamine is a common drug that's prescribed early in disease. You can see a difference in distribution of the gamma protobacteria. Obviously, the site of sample collection matters, whether you look at stool or biopsy, and then clearly antibiotics alter the distribution and abundance of these bugs. More importantly, if you look at ulcerative colitis and Crohn's disease, you begin to see shifts in your ability to metabolize short-chain fatty acids, your ability to make IL-10, and so forth, so that you begin to see differential distribution of patients who are healthy versus Crohn's disease and ulcerative colitis. And as the disease progresses in patients with Crohn's disease, you begin to see an abundance of the Enterobacteriaceae, more commonly in the terminal ileum of the gut. I also said that there are good and bad bugs. So there was a study by Harry Sokol that clearly demonstrated that Faecalibacterium propnitzii may be a good marker for disease. So if you have a recolonization post-surgery, the risk of recurrence was much less. But a word of caution here is that if you look at the Human Microbiome Project data, one of the bacteria that changed most abundantly, if your pulse and blood pressure changed, was also Faecalibacterium. So association to disease does not always mean causality. So, again, summarizing a paper that's recently been published in human genetics, unlike Mendelian disease, I'm going to show you shortly that complex diseases have many genes and many number of pathways. But on the context of the microbiome, again, it's likely that many microbes collaborate to cause disease. And one sort of take-home message from the initial analysis in adults was that you can see that the microbe community in IBD becomes more tolerant to oxidative stress. And some of the genes that are risk factors are, in fact, also in the same pathway, so suggesting that there might be communication on the microbe side and on the host genetic side. So I've tried to illustrate the concept of microbes in disease by using IBD as an example. But if you begin to look at some of the leading journals, there is clearly an expansion of the number of complex and metabolic diseases that are associated with similar changes in community structure and/or functional attributes. Obviously, most of our data today relies heavily on 16S sequencing and some metagenomics, which all of you are aware of the limitations of 16S. So to try to better understand what do we need. And in 2009, in a collaboration with Charles Mackay, we used a computational approach to identify GPR43 as a potential receptor for short-chain fatty acids. Follow-up studies went on to identify that this signaled into many different programs, not only epithelial injury and apoptosis, but more importantly, neutrophil chemotaxis, the production of reactive oxygen species, and cell death. So in a study that we've started with Michael Fishback, we've broadly asked the question, can we systematically answer the question of microbial profiles and functional output? So the approach that we've taken is to obtain microbial products and active biomarkers from microbes. At one point, when we can culture these individual organisms, we would like to do the same thing. And then in single well systems, ask the question, what happens to T cell activation, what happens to macrophage polarization, and what happens to epithelial cell commitment? And use both metabolomics and transcriptional profiling to begin to ask the question, how do these microbes and microbial products signal towards the host site? So back to the patient. So the disease in question is Crohn's disease and ulcerative colitis, and this is a colonoscopy appearance of a patient with severe Crohn's disease. So the ultimate goal of the lab is to now begin to understand what underlies that mucosal appearance. So what happens to the epithelium? What happens to Paneth cells and goblet cells? How are microbes sensed? How are the T cell amplification programs induced or suppressed? And how does the innate immune mediators that are recruited result in lymphocyte activation, transcription, ER stress, autophagy, and so forth? So a histological picture or an endoscopic picture needs to be translated into a molecular map, both on the host side as well as on the microbe side. And what I'm going to do in the next 5 minutes is to use autophagy as an example and try to show you some of the approaches that we've taken. So our interest in autophagy came about because of 2 genome-wide association studies, one in 2007 that identified ATG16L1 as a gene that's associated with risk of disease. And a year later, we identified a second gene called IRGM that is also associated with risk of disease. The 2 genes contribute to disease risk by different mechanisms. Very briefly, what is autophagy? It's a constitutive and conserved process that's present from yeast to human. It's active in every single cell, whether they be an innate cell or an adaptive immune cell or a cell involved in barrier function. There is an increasing number of disease associations with several of the autophagy genes. So in addition to Crohn's disease, there is now emerging evidence that some of the neurodegenerative diseases are associated with polymorphisms in autophagy genes. And we and others have more recently identified that some genes might be also associated with increased risk of TB. Autophagy is generally designed to do good, to heal inflammation, to control antigen presentation, and may even control lifespan. But obviously, programs in autophagy, when they are deranged or suboptimal, or hyperactive, one might result in disease. The autophagy pathway is complex. There are about 30 plus ATG genes that are conserved from yeast to humans that are essential for this program. And it relies, like the complement cascade or the clotting cascade, a sequence of multiple genes that have to come together to initiate this program. So there's one arm of an ubiquitin conjugation system that requires one of the initiator complexes, which includes ATG16L1. So ATG16L1 is a disease gene. To complete this double membrane, you need a second ubiquitin conjugation system that needs to come and grab your cargo. And this can be damaged organelles, it could be microbes, from viruses to bacteria, to parasites, and could be also protein aggregates. So I'll end there. All of this would not have been possible if not for an outstanding group of postdoctoral fellows and students in the lab. Obviously, the science has been accelerated by collaborations with my genetics colleagues, Mark Daly and David Altshuler. I haven't spoken to you about how we've used genetic insights to make small molecules so that we might be able to correct the disease with Stuart Schreiber. It's been a long-standing collaboration with Kip Virgin and Pat Steffenbach to begin to understand how autophagy contributes to disease and how multiple hits are needed to make disease. And then obviously I alluded to a very productive collaboration with experts in the microbiome field from Rob Knight, Curtis Huttenhauer, and Michael Fishback to begin to translate census or community structure to function, biology, and metabolite profiling. Thank you.
Dr. Xavier, and I particularly appreciated your indicating some of the technologies that we're going to need to really push the boundaries of this field. Question for Dr. Xavier, a clarifying question?
So that was a wonderful talk, and I have actually two short questions. One is, so recently, Lippincott-Schwartz has shown that the autophagosome membrane is derived from the mitochondria of the cell. And I'm wondering if in IBD you see changes in the numbers of mitochondria in the cells, or if you see changes in their activity. Obviously, you've got oxidative stress and things like that, but I'm wondering, in as much as we're now learning that, autophagy is really linked heavily to activity of the mitochondria. And the second thing is, could you define organoid?
So the organoid is sort of a structure that looks like the intestinal crypt, so the base of the intestine that has the intestinal epithelial cells, the goblet cell, the stem cell, and the Paneth cell. So this was a structure that was assembled initially by Hans Clevers, where he took the LGR5 positive stem cells and then isolated individual cells and put them together to recreate the base of the gut. And you can basically track these events through 3 or 4 different stages, where you get, you need cell-cell communication to build this structure. And what's emerging is that there may be more than one stem cell in the gut that is required. But clearly, the Paneth cell communication with the stem cell is critical to make a barrier that is efficient. With getting back to membranes and autophagy, so the emerging data is that there might be multiple contributors to the autophagosome membrane. And it could be mitochondria. There was a recent paper in Nature where ER membrane was thought to be important. There's also evidence that the plasma membrane is important. Clearly, the autophagy program regulates mitophagy, so the breakdown of mitochondria. We need to do additional studies to sort of begin to uncover how the autophagy mutations might affect mitophagy. But what's interesting is that, I think I briefly mentioned this yesterday in the evening, where we started to look at what metabolic circuits that mitochondria might communicate in response to innate signals. And one of the outputs is a cytokine called IL-1 beta that's made by a mitochondria-dependent program. We have a paper that's in press now where we've looked with Luke O'Neill to sort of begin to uncover how the mitochondria senses innate signals, differentially replicates, changes in size, changes in distribution, talks to other organelles, but results in the production of IL-1 beta in a succinate-dependent manner, which is totally unexpected, which is interesting because what I understand from the microbial community is that succinate is one of the key bug communication signals. So it might also happen within the cell system.
Thank you. So let's save further discussion for after the last speaker of this session.